Hi, grade 9. Are you fine? Today we are going to continue about solving exercises, supplementary exercise on digestion subject. Take this exercise, the effect of Coca-Cola on digestion. The first recipe for Coca-Cola, called French wine Coca, was invented by pharmacist John Pemberton States in 1885. At that time, Coca-Cola was then a drink advocated as a cure against gastric problems, including stomach pain and diarrhea. It was composed primarily of coca leaves, a medicinal plant from South America known for its digestive properties, and cola nuts extract with the stimulating and antidepressant qualities. Stimulating and antidepressant that act on the nervous system. It was supposed that Coca-Cola is favorable for the digestion of meat. To prove this hypothesis, we study the effect of cola on the digestion of protein. We performed the experiment summarized in the steps below. First, put in 3 test tubes labeled A, B, and C, 3 identical pieces of meat. The size and the mass of meat pieces are identical. The number of peptide bonds is 100 arbitrary unit in this protein. The peptide bond is made up between amino acids, 2 amino acids. You know that amino acids are the simplest constituents of protein. They are joined together by peptide bonds to form peptide, then peptide form protein, which is more bigger. Add to tube A water and pepsin. Add to tube B Coca-Cola and pepsin. Add to tube C just water. Place them in a water bath at 37 degrees for 24 hours. Pick up the tested hypothesis. Return to the text. It was supposed that Coca-Cola is favorable for the digestion of meat. So, the test hypothesis is Coca-Cola is favorable for digestion of meat. Convert the above text into schema using the legend, put, add, and place. In order to transform the text into schema, we use 3 test tubes, A, B, and C. Look at the meat. 3 identical pieces of meat are put in in both tube, and we add. Look at the add. This is the arrow that means put, and this is the arrow that means add. We add water and pepsin to tube A. We add pepsin and cola to tube B, and simply water to tube C. Place them in water bath. This is the water bath at 37 degrees for 24 hours. This is the legend, and we should not forget the title. Schema showing the conditions of the experiment. So this is how to convert the text into schema. If they ask to describe this experiment, in 3 test tubes, A, B, and C, we put an identical piece of meat, and we add water and pepsin to tube A, pepsin and cola to tube B, and water to tube C, and we place them in a water bath at 37 degrees for 24 hours. This is the description. The steps are given previously. After 24 hours, the number of peptide bonds was measured in the four test tubes. It decreases from 100 arbitrary unit to 80 arbitrary unit in A, while it decreases to 10 arbitrary unit in B, whereas in tube C, the number of peptide bonds doesn't change. Explain the decrease in the number of peptide bonds in the protein. The decrease in the number of peptide bonds means that bonds between amino acids that form the protein is broken and the protein is transformed into peptide. Do the results obtained validate your tested hypothesis, justify your answer? Yes, since in the presence of cola, the number of peptide bonds decreases from 100 to 10 arbitrary unit, while in the absence of cola, it decreases from 100 to 80 arbitrary units. Formulate a hypothesis that explains why the number of peptide bonds doesn't disappear in tube B at the end of experiment. Hypothesis: peptide bonds didn't disappear since the pepsin transforms the protein into peptide and not to amino acid. You know that the pepsin is an enzyme acting in stomach. In stomach, the digestion of protein occurs where protein is digested into peptide only and not into amino acid. This is the end of our video. Thank you for watching and goodbye in the next one.